Fantastic. Welcome to the stage, our next overgraduate finalist, Leiden.
Hi. In 2022, microplastics were found in human blood for the first time. Since then, a huge amount of research has come out detailing the negative health effects that this has, including embryonic developmental issues, cardiovascular disease, and even cancer. But how do microplastics get in your blood? How do they get in your body? Your food, your grains, your greens, your vegetables, they all contain microplastics. Taking a closer look inside of this wheat, microplastics. You eat this grain or this vegetable or anything that contains it, you also eat the microplastic. And they end up here through the use of plastic in agriculture. Mulch films and plastic-coated fertilizers are the main contributors to this problem. So if it's so bad, why don't we stop using them? Because we need them. Mulch films increase crop yield, support plant growth, and reduce the use of herbicides. Plastic-coated fertilizers, they allow for targeted release of nutrients, and they also prevent nitrogen volatilization, which actually helps us reduce the impact we have on climate change. As integral as these are, when they break down into microplastics. This pollutes the soil. And what's even worse is that these microplastics absorb toxins, heavy metals, pesticides. And so when you eat these, the combination of this has the adverse health effects that we talked about. Now, the EU also recognizes the major problem that this is and has now issued legislation demanding a change to biodegradable alternatives within the next 5 years. This is where PhaseOut comes in. Our project with PhaseOut, we use an innovative, sustainable feedstock, green methanol, which we feed to our bacteria that is able to produce the biodegradable plastic, PHA, that we then use in agriculture. So what's PHA? PHA is a biopolymer that's produced by a lot of microbes. It's usually produced for energy storage. However, what is so perfect for us is that it also has really similar characteristics to the conventional plastics that are used in agriculture. What's even better is that PHA is the only biodegradable alternative that ticks all of the boxes that have been deemed necessary by experts for an alternative. To highlight the key aspects of this, it can be made from renewable energy, it is biodegradable in all conditions, and it doesn't need arable land for production. However, this isn't the case currently. The conventional and current PHA production competes with our food security, has high CO2 emissions, and uses a financially unstable feedstock, sugar. This is the first problem of the PHA production process that we tackle with PhaseOut. What we do is we use green methanol as a feedstock. And we do this by taking renewable energy and turning it into green hydrogen, which we then combine with CO2 from direct air capture to make green methanol. After having spoken extensively to experts, we confirmed that not only is green methanol sustainable, but it's also future-proof. And by doing this, we create a PHA production process that not compete with our food supply. It uses a carbon-neutral feedstock and also a cheaper carbon source.
Thank you. So Ingrid has now told you a lot about methanol, but the question is, how can we actually leverage all the advantages of methanol? We do this by choosing our non-model organism, Methylobacterium extorquens, which can grow on methanol and already produces small amounts of PHA. The main goals of our process were to optimize the production of PHA and increase this and reduce the extraction costs, ultimately leading to a lower cost product. Since we are using a non-model organism, we heavily relied on modeling. We employed a genome-scale metabolic network model to identify key enzymes to either knock out or overexpress to then further increase the production of PHA in our organism. Initially, our model indicated that the PHB cycle, or in the PHB cycle, PHAC, or the PHA synthase gene, is a very promising target candidate. So our hypothesis was we overexpress this and we massively increase the PHA production in our bacteria. But since Methylobacterium is a non-model organism, the toolkit that we started with is quite poorly developed. So we started by characterizing a bunch of promoters by measuring M-cherry fluorescence. We identified PMXAF and PTUF as the most promising candidates to then continue with our overexpression of FAC. In the lab, we then found that when we overexpress this gene, we actually achieve a 24-fold increase in PHA production compared to the wild type. But we thought we can do a little bit better than that. So we went back to our model, which then also suggested that we could knock out some specific enzymes in the carotenoid biosynthesis pathway. The nice thing about this is that it not only redirects the metabolic flux towards the PHB cycle, but also knocks out the carotenoid pigments, which aid in UV protection, meaning we actually increase the biosafety of our germs because they are more sensitive to UV light, so we can easier disinfect equipment, for example. Again, here, we selected the CRT1 gene as our target and hypothesized that by knocking this out, we increase the PHA accumulation. We did, in fact, achieve a viable knockout of this strain. However, due to time constraints, we were not able to characterize it fully. Yet, the model suggested in combining this knockout with the PHAC overexpression leads to a 110-fold increase in PHA production over the wild type. So, I think we've now quite clearly shown that we can increase the production of PHA in our organism. So, on to our second part. We want to decrease the extraction costs. We did this by selecting 5 phase genes that we thought were compatible with our bacterium. The idea was to lyse the cells and then release the PHA as shown here in our animation. EJ1 and T4 lysins were the most promising candidates. We can see that we lyse almost all our cells in the culture, which results in the PHA release. With scale-up in mind, we decided to start prototyping with a small-scale bioreactor based on a Raspberry Pi, which is open source and low cost. And following this, we decided to also create an extensive entrepreneurship plan, spanning the proof of concept phase, the scale-up, and the production. We detailed very clearly how we want to acquire funds, invest and phase out, and make this a viable company. So, phase out aims to provide a cost-effective, biodegradable, and fully sustainable alternative for plastic use in agriculture. And with that, I would like to especially thank the sponsors that made this project possible, the team, and of course all the supervisors, and also iGEM and the iGEMers for making this such an enjoyable journey, making friendships and new memories. Thank you so much.